Hello everyone, welcome to my channel Online Biology Tutorials. In today's class, we will have a detailed account on AIDS whereby we would be studying about the discovery of the causal organism that causes AIDS. We will also study the structure of HIV, which is responsible for causing this disease. We will study how it passes from one organism to the other. We will study the life cycle of HIV. Thereafter, we will move on to its treatment. And in the end, we will say about its prevention. That's very important. We will be talking about the organizations which are associated with the prevention of the disease AIDS. So without further ado, let's get started with our discovery of the causal organism of this disease. First of all, the full form of AIDS is acquired immunodeficiency syndrome. Now, why are we using the word syndrome here? Because actually there is not one disease or one symptom in this disease. Rather, there are a collection of symptoms which are being noticed in this disease. So that's why we have used the word syndrome. Immunodeficiency is giving you an idea that in this disease, what is going to happen? There would be a deficiency in the immune system, because of which a person who suffers from AIDS will show various symptoms in their body because immunity is weak. Acquired means this is acquired, or a person suffers from it after the birth. In due course of their lifespan, they acquire it. That's why it is named as acquired immunodeficiency syndrome. Very important thing for you is, students, that you must know that the causal organism for this disease is human immunodeficiency virus, which is abbreviated as HIV. Means this is the causal organism or the pathogen which is responsible for causing AIDS. Okay. Now, first case of AIDS was noticed in the year 1981, and it was noticed in US. US means United States. However, in India, it came in 1986 and it was noticed in the Chennai, right? So, first case was noticed in Chennai. However, if we are saying globally, then first case was noticed in the year 1981. Now, next important thing, who isolated it? So, Montaguer, this is the name of the scientist, who along with his co-workers in France isolated this virus. Okay, so this is also important to learn, that Montaguer et al. In the France isolated this HIV that was responsible for causing the AIDS. Now let's move on to the structure of HIV, how it looks. Basically, this virus is an example of retrovirus. Now, who comes in this category? Retroviruses are a special class of viruses that in spite of DNA contain RNA as the genetic material. So we have already seen initially in the molecular biology that the flow of central dogma happens from DNA to RNA and then to the protein synthesis. But these viruses, they show the process of reverse transcription, means these retroviruses, they follow the process of reverse transcription to express themselves because they have RNA as the genetic material. So by the process of reverse transcription, they are going to form a complementary DNA, and this will ultimately form RNA, and then the proteins, they will be formed. Okay, so important thing to notice is that HIV comes in the category of retroviruses where RNA is the genetic material, and because RNA is occurring as the genetic material, so RNA will first form the cDNA and then flow of information will happen further, right? So moving ahead, you must know that there are two capsid in the case of retrovirus HIV. So these red layers are actually the capsid layers, and these capsid layers, as you know, they are made up of the proteins. So hereby you can make these, and this each circle is actually synonymous to the capsomere, right? Because capsid layer is made up of the protein, and you know that proteins are the polymers of the amino acid. So hereby each globular structure means a capsomere. And whenever we talk of aggregation of capsomeres, then we call this layer as the capsid. Okay, so capsid is made up of capsomeres, and there are two capsids which are being seen. Now in the center, two single-stranded RNA, they are seen, as I just said, that it comes in the category of retrovirus. So definitely it has to show RNA as the genetic material because it is single-stranded. So we can write either SS or we can write in full that single-stranded. Along with that, reverse transcriptase also found. This would be the enzyme that will do actually the process of reverse transcription. Okay, so reverse transcriptase is also found in the central region. Now another important thing for you is that outer to the capsid layer, envelope is found. Envelope is the outermost layer, and the glycoproteins on it, they are actually being derived from the host. So this is how a complete HIV looks like. You can see here that envelope has a glycoprotein on it. GP means glycoprotein. Glycoprotein is a conjugated protein in which, along with the amino acids, some carbohydrate part or carbohydrate moiety is also being attached, because glyco means carbohydrate and proteins, they aggregate together to form the glycoprotein. So here GP 120 is being written. 120 means the weight of it is 120 kilodaltons. Okay, so envelope has a glycoprotein on it and it is GP120. It is derived from the host. So you can see two capsids are there, single-stranded RNA molecules are there, reverse transcriptase is there, and of course on the outermost side envelope is there. Let's move on now to the transmission, how this HIV passes from the unhealthy to the healthy individual. Okay, so this comes in the title of transmission. So transmission basically happens through the sexual contact. Means if someone has AIDS and if that infected person will have sexual contact with a healthy person, of course AIDS is going to get transmitted. Other than this, transfusion of the blood is also a reason for its transmission. Means if the individual is infected with AIDS and that infected individual donates the blood to the healthy one, then this will lead to the transmission of this virus, which is HIV, and the disease AIDS will happen. The other reason is that it can also spread from the infected mother to the child in womb through placenta. So if a woman is suffering and she gets pregnant, then 100% chances are there that the disease will be transmitted to the growing embryo. And sometimes at certain places, syringes are used again and again. So if a syringe has been used to a person who is a sufferer of AIDS and this same syringe is used on a healthy person, of course HIV will going to get transmitted. So these 4 transmission reasons you have to learn. It can transmit through sexual contact, transfusion of blood from infected mother to the child through placenta, and of course through infected syringes. Now let's get on to the next title, that is the life cycle of the HIV. Now incubation period for HIV, it may range from 2 to 3 years to 10 years. Incubation period is a one whereby virus will invade the body and then it will show symptoms. Basically, in the initial stages, it affects the macrophages and then it affects the T helper lymphocytes. Okay, so first of all, macrophages will be infected and then the number of T helper cells will gradually decrease. T helper cells are the main cells of the immunity, which also helps in the humoral immunity, right? So if you have not watched my video on the humoral and cell mediated immunity, there I have discussed their role in the immunity. Kindly do watch it. Whether it would be infecting a macrophage or a T helper lymphocyte, the plan of action will be same. Okay, this red cell is our macrophage. We are drawing it as first because these would be the cells that would be infected. Okay, so let's say this is the single stranded RNA of the retrovirus, right? This virus will invade in the macrophage. This RNA has gotten. Now by the process of reverse transcription, RT means reverse transcription, it is going to form cDNA. Okay, it is going to form cDNA. Macrophages, they already have their own DNA. Now what would happen? This cDNA of the retrovirus will merge with the DNA of the macrophage, and when the DNA of the macrophage will replicate, these viruses, they will also increase. Ultimately their number will go on increasing and this will exert a pressure on the macrophage. Ultimately this macrophage, it will be bursted and these particles, these infectious particles, they will increase and they will be dispersed. Now, what would they do? Once they are released, they are going to infect the other macrophages. They will keep on doing it. So, gradually their number is increasing in the body. Now, normally when the incubation period is over, till that time, they already infect the T helper lymphocytes. Now, their plan of action in the T helper lymphocytes will also be same. Means the virus will go, it will do the process of reverse transcription, cDNA will form, it will merge with the DNA of the T helper lymphocytes, replication will occur, and the number of virus particles will increase in the body. So ultimately, when helper cells will be bursted or T helper lymphocytes will be getting infected by the HIV, then the number of T helper lymphocytes will be decreased in the body. Now, this is a very bad symptom for the body because actually T helper cells helps in the humoral immunity. So generally, the immunity will become less. When the immunity will become less, even by the passage of a small, really less toxic pathogen, the person will suffer because the immunity is weak. Cold may happen, recurrent fever may happen. So lot of symptoms will happen and a person will become sick again and again. So overall, immunodeficiency has to happen in the body, and that's why the name of the disease is acquired immunodeficiency syndrome, right? So this is how it goes on. So by the time tests are done and the person is diagnosed with this disease, enough damage has already been taken to the body. Now, what are the names of the diagnostic tests for AIDS? This is also important to know. One is ELISA, and the full form is also important. Enzyme linked immunosorbent assay. Immunosorbent assay. You have to learn its full form. It's important for you. And the other diagnostic test is Western blotting. Okay, so these are the tests that actually diagnoses that a person is suffering from AIDS or not. Now let's move on to our next topic. How is the treatment of this disease done? Now, very important thing, that the cure of this disease is not possible. So it's better to take some preventive measures through which the transmission of this disease may stop. Fine. So there are few antiretroviral treatments which are available, and these antiretroviral treatments, they actually drop down the activity of the reverse transcriptase. So reverse transcriptase is an enzyme which is found in HIV, and this will help in the reverse transcription. So once reverse transcription will not be done, cDNA will not be made. cDNA, when it will not be made, it will not merge with the host cells like macrophages or T helper cells, and ultimately its expression will not be done. Okay, but exact medicine for this disease is still awaited, and scientists, they are still working on it. Now, to move ahead and to sum up, there are various non-government organizations that are working for the control of this disease, and there is one organization which is known as National AIDS Control Organization. Okay, and it is abbreviated as NACO. Here NGO will become the abbreviation. So NGOs are the organizations which are run by the private organization. They are registered, but they are the non-government ones. There is an organization called NACO that is working on the control of the AIDS. They are just making the people aware that how its spread can be stopped. So they are just emphasizing people that disposable syringes should be used, because as I said few minutes ago, that because of using the syringes again and again, the disease will spread from unhealthy to the healthy individual. And sterilization of the dental equipments is also very, very important, because if they are not going to get sterilized, then the causal organism will pass from one individual to the healthy individual. And they also emphasize upon using the condoms so that sexual contact will not happen, fluids will not get mixed, and disease will not spread. Because as I said, there is no exact treatment available for this disease, so preventive measures should be taken. And the people in the rural areas, they are being emphasized that if anybody suffers from this disease, then they should not hide this thing, because this may further spread the disease. So if anybody suffers, then it should not be hidden, rather it should be known, so that that person can take all the essential measures to prevent the spread of this disease. I hope this was helpful to you. Please do subscribe to my channel, and if you like my video, do share it with your friends. Thank you. See you in next video. Happy learning.